Hi, today I'm going to be discussing the paper Design of Stapled Antimicrobial Peptides that are Stable, Non-Toxic, and Kill Antibiotic-Resistant Bacteria in Mice. The goal of this paper was to create a peptide that can act in place of an antibiotic to combat the increased threat of antibiotic resistance. So the researchers started with a peptide called MAG2, which they knew had antimicrobial activity, but it wasn't very stable and had a lot of red blood cell hemolysis and just other issues that wouldn't make it compatible with a living creature. So using this MAG2 peptide, they created MAG I plus 4 0 through 18 variations of it, which in which they stapled the peptide in different places in the sequence. This can be seen in table figure A, where you have MAG2 along with 0 to 18, MAG I plus 4 with their sequence, the antimicrobial activity for 4 different pathogens, as well as the red blood cell hemolysis percentage. So there's a very varying amount depending on where they put this. So in this process, they wanted to maximize the antimicrobial activity and minimize the hemolysis of the red blood cells. They found that the MAG I plus 4 15 variation was the best at this. As seen on the HNM for MAG I plus 415 on the right, there was no new HNMs, but there was increased activity and strengthened hydrophobic connections in the existing ones. Because of this, there was still a decent amount of red blood cell hemolysis. So they did further mutations seen in figure D on the right there, where they substituted K amino acid for all the ones in the sequence. And they found that A9 substituted with K created the least RBC hemolysis with the highest antimicrobial activity combination. So now this new MAG I plus 415 A9K variation can be seen in figure E, and that one has less hydrophobic interactions, which actually makes it a better candidate for this by decreasing the hemolysis even more. So then they decided to try another staple and staple it twice. So they stapled it in position 1 as well as 15 on the same MAG peptide, creating the figure at the top middle, B, where there was also very low hydrophobic connections and still has the A9K mutation. So then they inputted that into test mice or lab mice and found that that had even lower RBC hemolysis, very low kidney damage, which were like the main factors they were trying to minimize, but it still had an amount that was concerning a little bit. So they added some more mutations in order to stabilize the peptide more at B2, B21A, N22K, and S23K amino acids. So that was the final peptide they created in this study, for the most part, or the main one they created. And that ended up having an 88% success rate in curing the mice with these bacterial pathogens inside them. So it was pretty successful, and it laid the groundwork for more ways they could staple peptides in order to solve this issue. So thank you.